Hello, Rahman Rahim. Today we are going to discuss the small intestine. It is a site, you would say, is meant to complete your digestive process. Why? Because the nutrients are now supposed to be absorbed by its epithelial lining. Your small intestine is 5 to 7 meters long, and for this, I have to tell you that it is being divided into three segments. These are duodenum, jajanam, and ileum. Now you are looking into the cross-sectional view of the small intestine and the regular four courts of digestive system you are looking into. The innermost being the mucosa, then next is the submucosa, then you can visualize the muscularis layer, and finally the serosa is there. Now we have to learn about the internal surface modification of the mucosa. There are macroscopic as well as microscopic modification which we learn. In this very picture, you are looking into the surface modification known as plica circularis. What are they? These are the semilunar, you would say, or the circular folds present on the mucosa macroscopically, and they are made up of what? They consist of the mucosa and the submucosa core as well, and they extend almost about 1/2 to 2/3 of the luminal circumference. And why they are there? They are supposed to increase the surface area of your mucosa almost about 3 times. And where we get them? We find them most prominent in the jejunum, and we can see that they are entirely absent in the initial segment of the duodenum, and the ileum's distal half is also free of these plica. So these are the permanently folding of the mucosa visible to your naked eye, and is different from that of the rugae, which were temporary folds. And now histologically you can visualize that how plica appears. Plica looks like the outward extension, you would say, having the mucosa definitely and the core of submucosa in it. We have to reconsider that this is the plica circularis. I want to show you, and if you consider that the mucosa of the small intestine is being covered by another characteristic velvety appearance, finger-like projections, these are called as villi, which are 1 millimeter in its length. Now in this picture you are visualizing the villi again, and seeing that it consists of the outer surface epithelium made up of simple columnar cells, and inside there is a core of lamina propria, and what it contains, in addition to its regular loose connective tissue details, it has got the valvascularity, the arteries and veins you can visualize. And then another thing, important thing you can see in the center is the blind ending lymphatic capillaries, you would say. These lymph capillaries are called as lacteals. And what next? That the lymphatic nodules could be present in the lamina propria. And another important thing in that mucosa, that you have got the muscularis mucosae made up of inner circular and outer longitudinal. They are sending strands of muscular fibers towards the core of the villi, and they are meant for the rhythmic contraction of the villi, and that helps in the absorption process. The next to that villi, we will be discussing the another important surface modification. These are the crepts of libergans, or you call them as intestinal glands. They are present in between the villi, deep down into the tissue of the lamina propria of the mucosa. And how would you say that Their depth is almost 0.4 millimeter towards the mucosal surface as an invagination, and they are simple tubular glands which lie in the lamina propria. In the lamina propria, definitely will be addressing, in addition to the previous discussed features, the details of the connective tissue cells, and they are heavily infiltrated with the lymphocytes and other cells of the immune system, like plasma cell, you can mention macrophages, mast cells, and eosinophils. And we will mention that the places where these lymphocytes accumulate, largely they are supposed to form the lymphatic nodules or lymphoid follicles, and in some places like the ileum they become so big that they are supposed to form Peyer's patches, and they are supposed to extend from the mucosa into the submucosa. We will discuss them in detail later on. After all the details of the mucosa, we will mention the other feature as well, that there are the, I would show you again, that on the villi there are small minute projections called as microvilli, and these microvilli are basically present on the enterocytes or surface epithelial cells, which are simple columnar, and they possess the microvilli on their apical surface. They are also supposed to increase the surface area almost by the factor of 20, and we will visualize them under the microscope. So it's a microscopic modification. After that, we will discuss the submucosa. This submucosa is again showing what? That it is showing the loosely arranged connective tissue features, and it may be showing blood vessels, lymphatics, and submucosal or mesenteric nerve plexus. And what next? If we observe it in the duodenum, that will be addressing the Brunner's glands. We will discuss when we describe the detailed feature of the duodenum. And definitely we should know their production is of mucus, which should be having the alkaline fluid, I would say. Okay, next to it we will be observing the muscularis layer, which is muscularis externa, made up of inner circular and outer longitudinal layer. And definitely will be addressing in between the Auerbach nerve plexus, being a part of enteric nervous system. And on its very outer side there is a thin connective tissue layer called as serosa, and this serosa is definitely addressing the mesothelium, but at a few places like the posterior surface of the duodenum we can have adventitia over there. This is another very good view in which you can see that these yellow color projected upward parts are the villi, and their deep invagination towards the this mucosal surface from the surface epithelium are the intestinal glands. Now you can see, and these are the basal cut section of these simple tubular intestinal glands. This is important to visualize in this picture, and these are the neurovascularity and lacteals in towards the core of your villi. Up to so far we have learned that the histological appearance of whole small intestine is almost same, but with the few regional differences. Here is the slide, you can see is the duodenum, and towards the mucosa you can find the shape of the villi is like they are taller, broader villi, and appears as if they are of leaf shaped. And the number of goblet cells, which is the part of the surface epithelium, is going to be increased from the duodenum up to the ileum. They becomes maximum over there. And if you concentrate on its submucosa, we can see that the submucosa is addressing let me mark it with the ink. This is the submucosal glands, which are called as Brunner's gland. And they are what? They are branch tubular glands, and we what expect? They have to secrete mucus, which is alkaline fluid. They have to protect your duodenal mucosa against the acid coming from the stomach containing chyme in it. And the alkalinity of your Brunner's gland, they are supposed to bring the optimal pH in your intestine, the small intestine, and as a protective point, you would say, for the you would say the pancreatic enzymatic activity to be acted upon smoothly with the secretion of these alkaline nature Brunner's gland. The slide of the jajanam. Here you can see the taller plica and marked by the villi, and how they are appearing. These are the villi. These are the villi, and here they become shorter, narrower, and spressors, means they are lesser in number as compared to that of the duodenum. And if we see its the submucosa, you would say, we cannot find any type of gland over there that was initially present in the duodenum. Now here is the slide of ileum, in which what we visualize, we can see that there are basically, if you talk about the gross appearance of the ileum, definitely whole small intestine is having the two borders. One is mesenteric, other is anti-mesenteric. Mesenteric is addressing the attachment of mesentery and towards your posterior abdominal wall, and the other free border is called as anti-mesenteric. That would be addressing a special feature in its lamina propria, the larger sized lymphatic nodules, and they are called as Peyer's patches, and they are trying to enter into your submucosa. And over them you find that the villi are now lesser and sparest, you would say, shortest and narrowest rather. And prominent feature here are the Peyer's patches. And if we see the other mesenteric border, they would be addressing the somewhat taller villi. These are the few regional differences which we observe in the three parts of the small intestine. Now we have to learn about the surface epithelium and all the cells present over it. And as you know, the surface epithelium is covering whole of your mucosa and is being lined with the 5 type of cells. Number 1 are the enterocytes, which are the surface cells, you would say, and are columnar in shape. The other variety of the epithelium of small intestine are goblet cells. And then you expect there is a third variety present as a hallmark of small intestine are the Paneth cells. And we expect some enteroendocrine cells also a part of your epithelium. And of course the stem cells, which are undifferentiated stem cells made for the demand of mitosis. Now among the epithelial cells, we have to discuss one by one. We are supposed to look into the enterocytes, which are at their apical surface being lined with the small minute hair-like structure called as microvilli, and under light microscope their parallelly arrangement impart a striated appearance, hence the name brush border. They are looking like, and each microvillus is almost you would say 1 micrometer in length. And if you see the core of these microvilli, they carry 20 parallel actin filament, and just below the brush border the cell exhibits the terminal web, and this terminal web will address the transversely oriented cytoplasmic, you would say, filaments, and they are called as myosin filaments. What they do, they are present in the terminal web, and due to the interaction of the actin filament, which are present as a core in the microvilli, and the transversely running actin filaments, when they interact, they create oscillatory movement of your microvilli and facilitates the absorptive process. Another important thing, that under electron microscope, the tips of these microvilli, they are laden with the thick glycocalyx coat, and it has to protect the autodigestion of the mucosa. And they are well laden with various enzymes. They are supposed to hydrolyze the disaccharide and dipeptides into simpler one, the monosaccharides and amino acids, and then being absorbed by your enterocytes. And another important thing, that the enterocytes, they are having smooth endoplasmic reticulum at its apical region, and the basal region will be addressing the rough endoplasmic reticulum, and the cytoplasm is whole filled with the elongated mitochondria and large Golgi apparatus present as a supranuclear position. You can visualize the nucleus here. Each enterocytes, they are having the junctional complexes in between the two enterocytes or columnar cells. You can find here the junctional complexes. We have to see the cell which you already know for so many times are the goblet cells. What they are? They are having few microvilli towards their apical surface, you would say, and they are having the mucinogen granules in their apical sides, and they have got definitely the dilated top called as theca, you would say, and the narrow wine glass appearance having the narrow base, and this narrow base is addressing the nucleus in it, and all the regular organelles are concentrated near the base, like Golgi apparatus, the endoplasmic reticulum, which is a rough one, is abundant, mitochondria is there, and you find the nucleus being heterochromatic, and that is imparting the intense basophilic stain due to the presence of this nucleus, rough endoplasmic reticulum, and ribosomes. And when these mucinogen granules having the mucus content in it is supposed to be washed away by the H&E stain, you would say, the goblet cell looks empty. Again, now we have to discuss another variety of the epithelium, or could be the enteroendocrine cells, which are part of diffuse neuroendocrine system. They are producing different hormones, and which include cholecystokinin, gastric inhibitory peptide, secretin, and motilin. They are small basal cells you can find, and some other variety of the cell which we expect here are the stem cells, which are undifferentiated pluripotent stem cells made for the regeneration of the epithelium. Now we are supposed to talk about the Paneth cells, which are a hallmark of this intestine, small intestine. They are called as innate immunity cells, behave as natural antibody, and present where? At the bottom of the intestinal glands or crepes of libergans of the small intestine. They are showing intense basophilic staining at their basal region. Reason is the large, you would say, abundant rough endoplasmic reticulum and supranuclear Golgi complexes there. And their apical region is addressing or showing intense acidophilic stain, and this is due to the secretory granules present in that area. And what they carry? They have bactericidal agent called lysozyme and various proteins like defendants. Zinc is there, arginine rich protein, and they are all responsible for intense acidophilia of its secretory granules, and they have the ability to kill your bacteria and protozoa, so are supposed to play a very important role in regulating your intestinal flora, that is the bacteria which is normally present in the lumen of your small intestine. There is another variety of the cell we observe in the small intestine, which are the M cells, and these M cells you find over the epithelium of the ileum. I will show you next picture. Again, concentrate on another very important type of cells which are called as M cells or microfold cells. I would like to show you where they are present. They are present over the pyre patches of the ileum, on the epithelium that lies on the top of those pyre patches. In this epithelium, they are addressing the M cells. And if you concentrate on the shape of M cells, how they look like, they have got broad and flat dome-like apices. And what's special, the free apical surface is showing various microfoldings, and the basal region are definitely addressing various trenches, you would say, or deep pockets or recesses, and they are being occupied by macrophages and lymphocytes. Now we have to learn that how the antigen is approaching from your gut towards the epithelium, and of course have to interact with your M cells, which are now called as antigen transporting cells. Now what they do, they are supposed to endocytose these antigen and convey them to the, I mentioned, macrophages, and after being processed, they are supposed to reach towards the these lymphocytes, and these dendritic cells are mentioned here, which are the macrophages. And when these lymphocytes have interacted with the antigens, they are supposed to reach the regional lymph nodes after passing through thoracic duct, and they reach towards the bloodstream. And when into your circulation, the lymphocytes reach the lamina propria of the gut, and when they are supposed to reach over here, they are developed into the plasma cells. These plasma cells are supposed to secrete the IgA that reaches towards the lumen of the gut, and in this way the M cells are supposed to play important role in the immunological surveillance of the intestinal luminal antigen. You can see the course again, that the enterocytes are placed side by, the microfold cell is taking the antigen, endocytose it, and they are supposed to interact with the dendritic or macrophage cells. Then they come across to the helper T lymphocyte, you would say, B lymphocyte, then finally they are transformed into the plasma cells, and after that they are supposed to secrete, I mentioned, IgA, and then finally they are processed through and reach the lumen to cope with the bacteria.